variants are coming onto the map, and these are variants of concern. I just want to point out that viral variants are something that we're seeing all the time. I think a virus is really not a static entity. It's something that adapts constantly because it constantly makes little mistakes, and that leads to these changes, and then potentially the survival benefit for one variant, and then it emerges. So last year we actually had a variant that took over the whole world, but did not change much in terms of outcome, was not a variant of concern. Now we have variants of concerns that have started in the UK. And what England has seen is that they were really they went down with their numbers and at the end of last year, and all in a sudden they saw this shoot up of numbers, and also now registering a more severe disease, and that was the outgrowth of the UK variant. We have also variants now in South Africa and Brazil, and we're monitoring very closely a California or a group of California variants here. So next slide. So the UK variant has different numbers. You might see the Y501V1 nomenclature here. That's really one of the characteristic mutations that this virus has in the spike domain. Overall, it has 23 mutations, 8 are in the spike protein. And you might recall the spike protein is the protein that's on the surface of the virus here, giving it its very characteristic corona-like image. And is important for cell entry. And that is the target for the antibodies that are being generated by vaccination, or also antibodies that we can produce and give passively to people as a treatment or a prevention. Now, this new variant was first, described in the UK, has spread rapidly in Europe, and is now also in 31 states in the US. And there was a lot of questions what this 501 mutation is doing, but it's clear that it is probably helping the virus to enhance transmissibility. So the virus is between 40 and 70% more transmissible. So it is we have a much higher chance to get infected with that virus if we are in contact with somebody who carries it. And it is also, according to first estimates, 30% more lethal, especially in 60-year-old men or men over 60. There is now emerging tests coming out about how resistant, or is that virus resistant to the vaccine, or to people, or to immunity that has been arisen, but based on prior infections. It is about threefold more resistance to COVID-19 convalescent plasma, and only twofold more resistant to plasma from vaccinees that have received the vaccine. So it is not currently regarded as the main threat to inactivate the vaccine success, but it will be likely becoming predominant around the world, here in the US, very rapidly because of its rapid transmission. And I think we will have to really watch, how it affects disease. We know that in the UK, it also was infecting more, or was causing more disease in children. And I think these are all things that will have to be taken into account when we plan our march forward. The point is that there's first indications that is not a static entity. Again, I mean, this is currently 23 mutations that characterizing this variant, but it also acquires new mutations that could become more dangerous for the vaccine success in the future. So the next one is the South African one, which everybody watches with greater concern. It also has the 501 mutation, but it also has acquired these two other mutations, especially the 484 mutation, and you might have seen this in the literature. These are all in the RNA binding, in the receptor binding domain of the spike. So this is exactly the contact point between the virus and the receptor on the host cell, and that's what the antibodies are usually neutralizing, so that the virus cannot enter into the host cell. So this mutant is about 100-fold better in binding the receptor. What is more concerning is that these mutations, especially the 484, really confer resistance to antibodies. And we can look in monoclonal antibodies, which are sort of the Regeneron and Lilly ones that you can produce and then infuse in people. President Trump got it when he got infected. But we can also look again at the convalescent plasma, which is a mixture of different antibodies. And we know that it is 11 to 30-fold more resistant to plasma from convalescent from people who have had COVID. And it is about 6 to 8-fold more resistant to the antibodies produced after vaccinations. So I think it is not, it is still effective, but we sort of reaching a limit, and we're seeing that the vaccine has less effect here. It is now also in the US, as of a few days ago. It was first detected in South Carolina. But the point here is that people who have carried that variant have not had a travel history or contact with anybody in Europe or South Africa. I think it's clear that it is spreading in the community. And the point is that we have to be vigilant in terms of keeping all our measures in place to make sure that we prevent the spread of this variant in particular. I think there's a lot of discussions whether the vaccine has to be adapted, and I think the companies are responding. Especially Moderna has announced that they are working on an adaptation of the vaccine. Also BioNTech, I think, or an additional booster to raise the levels of antibodies. But what, as Warner has pointed out when he described the different vaccines, there is now trials that have been actually performed in South Africa, and so we have an idea on how effective these vaccines are against this mutant. And I would say the good news is that they seem to be all, or at least the ones that have been tested, effective against preventing death or severe disease. So the next one is the Brazilian one, also a 501Y mutant. You can see the theme here, but you also see that it basically has similar mutations like the South African one, the 484 and the 417. It's exactly the same one like we have seen in the South African one. So there's really a cluster of amino acids in the receptor binding domain that is variable and can mutate and become more efficient in binding and also evading the immune response. And this Brazilian variant really sprang onto the scene because it was first described in Manaus, in a city that had a very, very deadly first wave of infections, where 76% herd immunity was thought to have been achieved. And then all in a sudden, a second very deadly wave of infection was arising at the end of last year. And that was initiated by this variant. And so the idea was really that waning immunity in people who have been previously infected sort of is a fostering ground for this variant to arise and to circumvent this immunity. But it also sort of potentially speaks for the possibility of reinfection with these variants, even if you had COVID before. It is again very highly transmissible. We have talked about the mutations, and it has developed independently from the other ones, and we know actually from experiments from our colleagues that these variants can be selected for in vitro if we grow the virus in the presence of antibodies. It initially spread to Japan, but it's also been reported here in the US, and it's pretty clear that it is spreading in the US, and we haven't really fully found how much and where it is spreading. Just to re-emphasize, these variants are not static. They will continue to evolve, and sort of a second variant has already been identified in Brazil. And I think these are really countries where numbers are out of control, and it's a little bit the same thing for us. The more people are infected, the more chance the virus has to evolve variants. And that leads us to California. Unfortunately, our numbers are also not the best, as we know. This is a different variant. It does not have the 484 mutation, but it has this 452 mutation, which is very close to the other amino acids that I have been describing to you, and has been found actually in the mink virus that you might recall in Denmark that led to the slaughtering of many minks. And so this has been also observed in vitro and in vitro experiments that this mutation can arise in the presence of antibodies. So we're watching this with great concern. It also appears to be highly transmissible. I can jump down here. It was responsible for the high attack outbreak at Christmas in the Kaiser ER, where there were 79 people infected. And we are currently growing this virus in our BSL-3 to test whether it is neutralized by antiserum from vaccinated or convalescent people. And I think there's a large consortium here in California forming to really get ahead of these variants with great sequencing and to make sure that we really know how effective they are, how effective the vaccines are against them, and also to especially see how they continue to mutate and if further mutations here, specifically the 484, might be acquired. So in summary, I think we really have to make sure that we stop transmission of the variants and the whole virus. I mean, we have to really bring our numbers under control, because otherwise these mutant viruses will become dominant and ultimately could undermine vaccine effectiveness. I think we are a little bit in a race between the vaccine and the variants. I think really, again, it's nothing that surprises us as virologists, because the virus is doing what the virus is doing. It adapts. And I think we need to make sure that we now roll out the vaccines as fast as possible to bring down the rate of infections and to make sure that the variants cannot further foster. And yes, I think we need increase our overall surveillance. And I think we have to adapt also our therapeutic monoclonal antibodies. You have heard about the Regeneron cocktail. I think this all has to be rethought to make sure that we're not giving the wrong antibodies. And I think we have to make sure that we adapt also our vaccines. Multivalent RNA vaccines are very easy, relatively easy to achieve. And I think the way how the companies are responding is very positive.